Hello everyone, Namaste. I'm Gautam Prasad Chaudhary. Welcome to my channel. And today we are here with a new topic about the anatomy of a excretory system. So today we will talk about this topic in more detail. And this excretory system plays a very crucial role in maintaining hemostasis by eliminating the waste product from the body. And the primary organ involved are the kidney, ureters, urinary bladder, and urethra. So let's begin this topic in more detail. So you can see in my next slide, this is the structure of a kidney. You can see this is a two pair of a kidney. This is a one pair of kidney, and there are one pair of ureter and one bladder and one urethra. So one by one we'll discuss each and every structure of a kidney. So let's begin first structure of a kidney. If you see, the human body has two kidneys, and these kidneys are a pair of a bean-shaped organ located on either side of a spine, just below the rib cage. And each kidney is about 10 to 12 centimeter in length and 5 to 7 centimeter in width, and its thickness is about 2.5 to 3 centimeter. And the average weight of a kidney is about 120 grams to 150 grams. And one interesting thing is that the right kidney is slightly lower than the left kidney due to the presence of a liver above the right kidney. So if we see the two kidneys, the positions, the right kidney is slightly lower than the left kidneys. And this is one important thing about the kidney. So let's see the structure of kidney in more detail. So if you will see the structure of a kidney, you can see the outer layers. This is known as a renal capsule. This is outer covering is known as a renal capsule, and the capsule is a very taut, fibrous and outer layer that protects the kidney. And if you go below this capsule, you can find the outer regions. This is a cortex, and this outer region containing nephrons, and these nephrons are the functional unit of a kidney. And below this cortex, if you go more depth, or if you go more inside the kidney, you find a medulla. The inner regions, you can say the inner regions of a kidney is known as a medulla, and it is made up of renal pyramids which contain collecting ducts. So this funnel shape like structure, you can see these are known as a renal pelvics, and this structure collects the urine before passing it to the ureter. So also there are many millions of nephrons, and these nephrons are microscopic structures which filter the blood and regulates the water and electrolytes, and it maintains a water and electrolyte balance. And each nephron consists of a glomerulus. Along with the glomerulus, there are Bowman capsule, proximal tubules, loop of Henle, distal tubules, and a collecting duct. So this was overall introductions about a kidney. And if we'll see the function of kidney, the very first important function of kidney is that the kidneys play a very important role in the filtration of blood, which remove the metabolic waste product like urea, creatinine, toxins. Also, the kidney plays a very important role in regulation of electrolyte balance like sodium, potassium, calcium. This kind of electrolytes are being balanced by the kidney. And number 3 function of a kidney is that it controls the blood pressure through a renin-angiotensin system. In some other chapter we'll discuss about this RAA system. And apart from this function of kidney, we can also find that the most important function of kidney is that it plays a very important role in production of erythropoietin. And this erythropoietin is a hormone that stimulates the red blood cell productions. And finally, this kidney plays a very important role in regulation of acid-base balance. So let's see the next structure of our excretory system, that is ureters. And you can see two ureters from each kidney. From left kidney, one ureter is coming down, and from right kidney, one ureter is coming down. And these ureters are a thin muscular tube, about 25 to 30 centimeter in length. They play a very important role in transport urine from the renal pelvics of a kidney to the urinary bladder. The ureter have 3 constituents. Number 1 is the uretero-pelvic junctions, and these junctions where the ureter meets the renal pelvics. And if you come below, you'll find a crossing over the iliac artery near the pelvic brim. And next junction is the uretrovesical junctions, where the ureter enters the bladder. So from where they are coming out and they are meeting the renal pelvics, they are called as the uretropelvic junctions. By name itself you can identify the junction, and the last junction is the uretrovesical junctions, where the ureter meets into the bladder. So these are the three constrictions. And if you see the wall of a ureters, they contain a smooth muscles that help in propelling the urine through the peristalsis, preventing backflow. So this was a second structure. If you will come below the ureters, you will find the urinary bladder. So there is only one bladder for male and female. There was two kidney, there was two ureter, but there is only one bladder. So the urinary bladder is a hollow muscular organ, and that temporarily stores the urine before the excretions. So whatever the urine are produced from the kidney, they are coming down and they are stored in the bladder, and this bladder are located in the pelvic cavity. You can see in the figure, and behind the pubic symphysis, the bladder are distorsor muscles and expand as it fills with the urine and contracts during the urination. So they have a property of contractions and relaxation. They have that kind of a property. So if you see the anatomical features of a bladder, you'll find a flowing regions, you will find a trigon. These are triangular areas found by opening of two ureters and a urethra, and also you can find a rugae, that is a fold in a bladder wall that allow the expansions. And similarly you will find the internal and external sphincters that control the release of a urine. The internal sphincters is involuntary in nature, while the external sphincter is voluntary in nature. So if you see the capacity of bladder, they can hold up to 400 to 600 ml of urine before the urge to. So this was about the male urethra. But if you see the female urethra, they are little bit short compared to the male. They are about 3 to 5 centimeter in length, and there was two kidney, there was two ureter, but there is only one bladder. So the urinary bladder is a hollow muscular organ, and that temporarily stores the urine before the excretions. So whatever the urine are produced from the kidney, they are coming down and they are stored in the bladder, and this bladder are located in the pelvic cavity. You can see in the figure, and behind the pubic symphysis, the bladder are distorsor muscles and expand as it fills with the urine and contracts during the urination. So they have a property of contractions and relaxation. They have that kind of a property. So if you see the anatomical features of a bladder, you'll find a flowing regions, you will find a trigon. These are triangular areas found by opening of two ureters and a urethra, and also you can find a rugae, that is a fold in a bladder wall that allow the expansions. And similarly you will find the internal and external sphincters that control the release of a urine. The internal sphincters is involuntary in nature, while the external sphincter is voluntary in nature. So if you see the capacity of bladder, they can hold up to 400 to 600 ml of urine before the urge to urinate is felt. So below the bladder there is only one urethra, and that is for female. If you see the female urethra are a little bit short compared to the male, and so there is a more chance of getting the UTI infections in a female. So the urethra is a final passage through which urine is expelled from the body. It varies in length between the males and females. If you see the male urethra, they are about 18 to 20 centimeter long, but this length are maybe different in some books, but the average is around 18 to 25 centimeter, but in some books they have written as a 25 centimeter long. And these male urethra are divided into three parts. They are urethra, membranous urethra, and spongy urethra. So this prostatic urethra are those urethra that passes through the prostate gland. So if you will come below the prostatic urethra, you will find a membranous urethra. They are shortest and narrowest part of the urethra. And also you will find a spongy urethra that run through the penis and open the external urethral orifice. So this was about the male urethra. But if you see the female urethra, they are little bit short compared to the male. They are about 3 to 5 centimeter in length, and because of a short urethra, female has a more chance of getting a urinary tract infections due to its shortest length. Okay, so this was about urethra. So we have already talked about kidney, ureters, bladder, and urethra. So in conclusion, we can say there are two kidneys bean shaped, and they filter the blood and produce the urine, which is transported by the ureters to the bladder for storage. And when the bladder reaches its capacity, urine is expelled through the urethra. And this system plays a vital role in maintaining the fluid balance, and it will play a very important role in removing the toxins and regulating the blood pressure. So understanding this anatomy of excretory system help us to appreciate how our body efficiently manages the waste eliminations. So we'll see the next topic of today's class. So we'll try to cover as much as possible because we don't have much more time. Now we'll talk about urine formations. So as far we have studied various aspects of excretory system. So the major organ in excretory system is a kidney, as we know, and although other organ also involved, the kidneys plays a very important, the most important role in excretions. And the kidney removes the waste product produced during the metabolic activities in the body. So we have this discussed in the previous slide. So inside the kidney there is a functional unit that is responsible for this process, which is known as a nephron. So if we define the nephron can be defined as the structural and functional unit of kidneys, and each kidney contain approximately 1 million nephrons, and the nephrons plays a very important role in filtration of blood, and they remove the waste product and retain essential substances that is essential for our body. So already we have discussed about the structure of the kidneys, so no need to talk about these things. So let's talk about the blood supply to the kidney. So we can find a renal artery. You can see this area. This is the renal artery. This artery brings the blood into the kidney. So inside kidney, this artery branches into the smaller blood vessels, which eventually reach the nephrons. So also you will find the renal veins, and this renal veins carries a filtered blood away from the kidney. So this is a difference. Renal artery brings the blood into the kidney, whereas the renal vein carries a filtered blood away from the kidney. So this was about the some missing structure of a kidney, and let's talk the nephron structures in more detail. So if you see the structure of nephron, so this is the structure of a nephron, you can see on my screen. So you can see many parts, like here you can see this is a PCT, proximal convoluted tubules. Similarly you will see here loop of Henle, and loop of Henle has a two limb. One is a descending limb and next is a ascending limb. So this is a descending limb and this is a ascending limb. So here you can see DCT, distal convoluted tubules, and finally you can see here collecting duct. So let's see what happens to this part. So if you see in this proximal convoluted tubules, in this part maximum reabsorption happens. Here about 99% of water are and essential fluid are reabsorbed, like glucose, amino acid, sodium, potassium, bicarbonate are reabsorbed in this PCT part of the nephron. So the broad border epithelium, you can see here there are broad border epithelium that increases the surface area for the maximum absorption. So these are present in the PCT, proximal convoluted tubules. So this is a loop of Henle, as I earlier told. So there are two limb, one is descending limb and next is a ascending limb. So in descending limb, this limb is permeable to the water but not to the ions. That means water moves out, making the filtrate hypertonic. But if you see the ascending limb, these are permeable to the ions like a sodium and chlorine, but not for water. That means they removes the salt, making the filtrate hypotonic, not hypertonic, making a filtrate hypotonic. Okay, so this was about a loop of a Hanel. So if you go a little bit above, you will find this is a DCT. So this in DCT part there will be no reabsorption of so in this part what happened, there will be regulated reabsorptions, means controlled by the hormones like aldosterone and ADH, and also there are the calcium and water reabsorptions and depend upon the body needs. So the final part, that is a collecting dart. So in this part what happened, there is a final water reabsorption, so under the control of ADH, antidiuretic hormones. So I'll talk about ADH in next slide a little bit. What is the role of ADH, how it play a role, important role in absorption of waters or diuresis. I'll talk in next slide. So also in this collecting dart, what happens, there is a urea is reabsorbed that helps in maintaining the osmolarity. So this was a part of neurons, and what happened in this part, like in PCT what happens, in a loop of Hanel what happened, in DCT what happened, in collecting dart what happens. So this was all about nephrons and absorptions at different parts. So now let's talk about the urine formation. So this is an important topic of today's class. So the process of urine formation is essentially known as a blood cleansing process. So blood cleansing process means refers to the removal of unwanted substances from the blood, ensuring that the elimination of toxins. So this filtration process takes place in the kidney, which is responsible for removing the waste product while retaining the essential substances. So we have already discussed about the structure of kidney and nephrons. Now we'll focus on the urine formation in more detail, how blood is filtered inside the nephron to remove the unwanted substances while retaining the useful ones. So this is the important and interesting topic us. So focus on our topic. Okay, so let's see the steps of urine formation. So there are mainly 3 steps for urine formations. Number 1 is glomerular filtrations, number 2 tubular reabsorptions, and number 3 tubular secretion. That is a filtration in the glomerulus and reabsorption and secretion. So these 3 steps take place within the nephron, which we have already studied. Now let us examine how this process works. So number 1, let's talk about glomerular filtration. So this glomerular filtration occurs in a glomerulus. So glomerulus are a network of capillaries located within the Bowman capsule. So blood enters the nephron through the afferent arterioles. You can see in this figure, this is a afferent arteriole and this is a efferent arteriole, afferent and efferent arteriole. So this afferent arteriole which branches into the capillaries forming the glomerulus, and the blood then exits through the efferent arteriole. So if we see the diameter of this arteriole, we find that the afferent arteriole has a wider diameter than the afferent arteriole, and the pressure inside the glomerulus increases and leading to the filtrations. This was about glomerular filtration. So if when blood enters here, the blood contains plasma and some kind of cellular components like red blood cells, white blood cells. So during filtrations, only plasma is filtered into the Bowman's capsules, while the cellular components remain in the blood, right? And the filtrate consists of water, glucose, amino acids, sodium ions, and some other molecules, but proteins are not filtered because of their large size. So this process is called ultrafiltrations, as it occurs in a high pressure regions. I have told why the pressure is more, because the diameter of afferent arterioles has a wider diameter than the afferent arteriole. So the filtrate fluid now called as a glomerular filtrate and moves into the proximal convoluted tubule. Okay, so next step is a tubular reabsorption. So after glomerular filtration, the filtrate consists of several essential substances that body needs, and these substances must be reabsorbed back into the bloodstream. So this process occurs in different part of a nephron, mainly in proximal convoluted tubule, loop of a Henle, and a distal convoluted tubules that I have discussed in a structure of a nephron in earlier slide. So in PCT, we can see that majority of reabsorption occurs here. Useful substances like glucose, amino acid, sodium ions are actively transported back into the blood. And next is a loop of Henle. Here you can see. So in the loop of Henle, this help in a water and sodium balance by reabsorbing the water and sodium ions, so which is crucial for maintaining the blood pressures. So next is a DCT. So further adjustment in a ions balance take place here. So now let's talk third steps, that is a tubular secretion. So in this tubular secretions, this is a final step in urine formations. So some substances that were not removed during the filtrations need to be actively secreted into the tubules for excretions. Substances like hydrogen ion, potassium ion, and certain drugs are secreted from the blood into the tubules, and this helps in maintaining the pH balance and electrolyte balance in the body. So this was about different 3 steps for urine formations. Now let's talk about the excretions, how this urine excretion occurs. So after this 3 process, the urine collect in the collecting ducts, which transported it into the renal pelvis, and from where it moves to the ureter and then bladder, and finally excreted through the urethra. So you can see in this figure, from here it goes to renal pelvis, and from renal pelvis it goes to the ureter, then finally they are collected in the bladder, and they are passed out, they are excreted through the urethra. So let's talk about GFR, that is a glomerular filtration rate. What is this? So GFR is the rate at which blood is filtered by the kidney. So here the total cardiac output, that is about 5 liters per minute, that the blood pumped by the heart per minute is 5 liters per minute. So about 20 to 25% of this blood is directed to the kidneys, meaning the 1100 to 1200 ml of blood per minute is sent to the kidney for the filtrations, and GFR is about 125 ml per minute, so leading to approximately around 180 liters of a filtrate per day. So here is one interesting thing, that your GFR is about 125 ml per minute. If you look at it per minute, it's interesting, but how much urine is produced? Only 1 to 1.5 liter. How much? Only 1 to 1.5 liters in 24 hours. So how this is happening? Because the plasma is being filtered and water is also being removed. However, most of this water is reabsorbed. If all the water were excreted, it would lead to dehydrations. So body reabsorb it, and that's why the final urine volume is only about 1 to 1.5 liters per day. This is the reasons. Now let's understand the renal blood flow. So how much blood reaches the kidney per minute? So if we see, renal blood flow is approximately 600 ml per minute. So this is an important question. From this renal blood flow, the filtration rate is determined, which tell us how much blood is filtered at the glomerulus. So there is one formula how to calculate the filtration fractions. So filtration fractions can be calculated by GFR divided by a renal blood flow, which is approximately 19 to 28%. So now let's understand the ultrafiltrations. What does ultrafiltration means? It means filtrations due to the pressure. The pressure in the glomerulus forces the blood plasma to filter. However, the protein are not filtered in this process. All right. As I told that function of aldosterone. So what is the function of aldosterone? So what does this aldosterone facilitate the absorption of? So it facilitates the absorption of NaCl. Who facilitates the absorption of NaCl? Aldosterone. And who facilitates the water absorption? Again, the answer is ADS, antidiuretic hormones. So now consider the condition like diabetes insipidus. In diabetes insipidus, what happens if there is a ADS deficiency? So in these conditions, ADS is not able to reabsorb the water, leading to conditions known as diuresis. This is why the ADS is essential for the water absorptions. So this is the important role played by the aldosterone hormone in the kidney. So next slide is urination process or maturations. So for maturations or urination process, we have 5 steps. What happens? First is the bladder filling. So whenever the urine are produced from the kidney, they moves into the bladder via ureter. So once there is a complete filling of bladder, what happens in bladder? There are some receptors. So these receptors will detect when the bladder is full, and they send us signals to the central nervous system. So once the central nervous system get the message that the bladder has been full, the CNS trigger the muscles contraction in the bladder wall. So once the there is a muscle contractions, there is a internal urethral expander, which is involuntary in nature, that relax automatically. And also there are the external urethral expander, that is voluntary in nature, and that can be controlled to delay the urination if it is needed. So this is how the urination process occurs, and it is a very important that you should not hold your urine for a long time, because if you hold a urine for a long time, it can cause infection or bladder issues. So guys, this was all about the excretory systems. We have discussed about the anatomy of excretory systems. Also, we discussed about the structure of nephrons and different site of absorptions, and also we have discussed about the formation of urine, and also we have seen about the urination process. So this was all about today's class. Thank you for watching this video. Bye bye. Have a good day.